My name is David Otaegi. I am head of the multiple sclerosis unit in the Instituto Biodonostia in San Sebastian, Spain. Our work is focused in the expression of the genes in blood of multiple sclerosis patients. So in the different stages of the disease, multiple sclerosis is a disease that have attacks, and after the attack the body tries to remyelinate the brain. We are trying to understand how that process works to help the body to do the remyelination and to improve the quality of the life of the patients. We study the DNA of the patients to see the difference with the DNA of the healthy people, searching for polymorphisms or haplogroups that could give susceptibility to suffer the disease. And the question is try to understand how the treatments in the disease works, how these treatments are affecting the expression of the genes. If we can find a biomarker that helps the clinicians to manage the different treatments. We are pretty concerned about the regulation of the net of the expression, and for that we are very interested in microRNA, that are tiny molecules that could have a very powerful effect in the expression of the genes. So we are trying to understand how the microRNAs are working or which role is playing in the MS. When we think about the project, we think a lot about which is the best platform to do it. Also, this project is in collaboration with other groups, so we have to talk with other groups and try to choose the same platform. So we think about arrays, we thought about deep sequencing, but finally, for this project, for search biomarkers, we thought that the best platform will be Tagman assays. Tagman assays give us the opportunity to validate quickly the data with microRNA that are validated in the literature, and also the assays are validated. So we know that the data that we have, we can believe it, but I like Tagman. It's specific. I don't have to think about it. I don't have to be worried about it. I have to worry about the data. So QuantStudio gives us the opportunity to measure the microRNA profiling with Tagman assays. I was working with quantitative PCR all these years. I work with low-density arrays for studying the microRNA profiling. We need to make more pools. Now in the QuantStudio we can do in 2 pools all the platform. We have also the AQ field that dispense the sample in the open array, so the preparation of the sample is fast. As we start the work and the study of the profiling with Tagman assays, we hope that the validation will be faster because we are going to use the same Tagman assays in another way in the same machine. So that's one of the main barriers that we can solve with this machine. QuantStudio is a small machine. Using few space of the bench of the lab, you can have all the blocks, so you have more space for the other machines. Gives you the opportunity of run different kind of studies because you have these different kind of blocks. So in a not so big machine, you can run a lot of experiments. We can play with the several blocks, so I can have the profile with the open array system. I can validate it with the 384 plates. I can work with SNPs. Maybe in the future for virus I can work with digital PCR. For the genomic units, QuantStudio will be a good platform because it gives you a lot of versatility. Not every group needs all the blocks, but if you want to be a facility for all the researchers, all the institutions, you need to give the opportunity of work with all the blocks. If I want to work with biomarkers, and I know what I am looking for, and I want to validate, or I want to be pretty sure that the microRNAs that I am looking are validated and are strong, I will work with QuantStudio. If I don't know what I am searching for, and I am looking without any hypothesis, I will work with Ion Torrent. I will have all the small RNAs, almost. Also, I will have the sequence. That will be very important. If I have to make a workflow, I will study the expression of the microRNAs, in this case with the microRNAs, with ion torrent. I will check or validate it with Tagman assays. I am happy with the Life Technologies products. I work with it a lot, with the sequencing product, with the quantitative PCR products. I enjoy the support of our team in Spain. I think this is one of the strongest points of Life Technologies.